class. Okay, good day, class. So for our meeting today, we're going to tackle Lesson 3, Secondary Hemostasis. Now, secondary hemostasis class involves the generation of sufficient thrombin to convert your fibrinogen to the fibrin clot. Now, if you try to compare this with your primary hemostasis class, the primary hemostasis, if you still remember, would include the blood vessels, or the vascular intima, and the platelets. But for the secondary hemostasis, it basically involves, if you're familiar with the so-called uh, coagulation factors. And the main goal of this hemostasis is to convert or to produce enough thrombin to convert fibrinogen to a fibrin clot. Because remember, class, if you suffer from a injury, the end product should be the formation of a stable fibrin clot. Now, the secondary hemostasis would involve a series of interaction of coagulation proteins leading to the formation of a stable fibrin clot through activation of intrinsic, extrinsic, and your common pathway. Now, with the generation of thrombin, there are two stages: the initiation and the propagation. Simply put, class, if you try to understand these two words, initiation is the start of the conversion of your fibrinogen into your fibrin by thrombin. So that's initiation. Initiation is the start of the conversion of fibrinogen into fibrin by thrombin, while propagation is the increase of uh, of thrombin, or increase in the number of thrombin, class, leading to more conversion of fibrinogen into fibrin. Now, let's talk about the plasma components of the coagulation system. Now, one thing you need to remember here are the zymogens. Now, zymogens, class, are a type of proenzyme. When you say proenzyme, they are technically not enzymes. These are considered inactivated enzymes. They would circulate in the plasma, and they are inactive and becomes activated only if there is a tissue injury. So again, class, your proenzymes or your zymogens are inactive enzymes. They are circulating in your plasma. They are inactivated and would only be activated if there is a tissue injury. Thus, if there is no tissue injury, class, they are just present in the plasma as zymogens. Now, cofactors will only aid or help in the function of the zymogen. So remember this, cofactors, class, they basically aid or help in the function of zymogen. Now, HMWK, tissue factor, factor 8, and factor 5 are involved in the coagulation pathway. These are cofactors, class, for zymogens in the coagulation pathway. Protein Z, protein S, and thrombomyogulin are cofactors for control protein. These are substances, class. They are called control protein because they regulate our coagulation system. So remember the three: Z, S, and thrombomyogulin. They basically regulate our coagulation system. Now, I want you to remember this, class. How would you know? How would you know if a coagulation factor is activated? We always add the lowercase a, or the small letter a, after the numeral. For example, class, factor 7 alone is considered an inactive coagulation factor. But if you add the small letter a or the lowercase a there, it becomes active. Another example, factor X alone is considered inactive. For it to be activated, class, you just add the letter A, the small letter A. So do remember that. Now, here are two tables, class. Please remember them and do memorize them. They are the plasma procoagulant serine protease. Here we have the inactive zymogen and the active protease. Basically, this is just a conversion of an inactive zymogen to become an active coagulation factor. So if you take a look here, yung mga active coagulation factor have the small letter A in them. Then they also have cofactors class that help in the aid in the function of the zymogen. Then they have substrates, so do memorize them. Then we also have the hemostasis cofactor. So we have the different cofactors, your tissue factor, 5, 8, high molecular weight kininogen, thrombomodulin, protein S, and Z. Then they have different functions, and they can also bind. Please take note of this. They bind to different coagulation factor. An example here is that your tissue factor would bind to activated factor 7, while your cofactor 5 would bind to activated factor 10. So again, do memorize these two tables. Now, what is the nomenclature of your coagulation factor? Now, this was the nomenclature or the naming system was discovered or was implemented in 1958 by the International Committee for the Standardization of the Nomenclature of the Blood Clotting Factors. Name the procoagulants in the order of their initial description or discovery. When activated, lowercase a appears behind the numeral. Both zymogens and cofactors would become activated. Now, some factors are customarily identified by name rather than Roman numeral. So technically, class, ang mga coagulation factors, your coagulation factors are named using Roman numerals. But there are certain factors that would use the normal name. Example would be fibrinogen, which would be factor 1, prothrombin, factor 2, tissue factor, factor 3, ionic calcium, factor 4. Without Roman numeral designation are your platelet factor 3, prekallikrein, and your HMWK. So remember this class. Those without coagulation factors without Roman numeral designation are your PF3, PK, and HMWK. Now, I want you to memorize this table, class. I won't go through them anymore. This is very easy to understand, very easy to memorize. Do take note of the different coagulation factor, their names, their function, their molecular weight, their half-life, and their amount or concentration in the plasma. Now let's go to the biochemistry of coagulation factors. Now your coagulation factors class are proteins class. They are all proteins produced in the liver except factor 3, which is produced in the tissue, factor 8, which is produced in the liver and endothelial cells, platelet factor 3 produced by the platelets, von Willebrand factor produced by endothelial cells and platelets. So all of your coagulation factors class are produced in the liver except for these four. Factor 3 in the tissue, factor 8 liver and the endothelial cell, platelet factor 3 by the platelets, and von Willebrand factor endothelial cell and platelets. Now 8 of these coagulation factors class are proenzymes, procoagulants, or zymogens. They are inactive form, which are either. So they could become activated or inactivated. So again, 8 of these coagulation factors class are either inactive form or inactive form. While 7 serine proteases. So these are already activated class. Your factor 2A, 7A, 9A, 10A, 11A, 12A, and your kallikrein. Then we have your transglutaminase or your factor 13A. So this 8 class are the 8 that could either be in active or activated form. 7 of those are serine protease and 1 is a transglutaminase. They have no biologic activity unless converted to active enzymes. Now take note class that serine proteases are proteolytic enzyme of the trypsin family. They have a reactive seryl amino acid residue in their active site. Action on substrates, they are able to hydrolyze peptide bond synthesized as inactive zymogen consisting of a single peptide chain. Activation class is done by cleavage at one or more specific site by another protease. hydrolysis of peptide bonds. So these are their activation. So your serine proteases class become active by cleavage at one or more specific site by another protease or hydrolysis of peptide bonds. Now, four of your coagulation factors class are cofactors. Basically, they help or they aid in the function. These are your tissue factor, factor 5, factor 8, and HMWK. Now, tissue factor class is located on the membranes of your fibroblasts and smooth muscle cells, while factor 5, factor 8, and your high molecular weight kallikrein are soluble plasma factor. They are non-circulating protein of the subendothelium. They are able to bind, stabilize, and enhance the activity of their respective or particular serine protease. So they are able to bind, stabilize, and enhance. Coagulation factors gain stability and increase the activity with the help of these cofactors. Now let's talk about the summary class. So this is like the summary. Now the members of the kinase system would include your HMWK and your perikallikrein. Now tissue factor is located in the lining of the blood vessel layer. Now I want you to remember this class, this one. Soluble plasma factors. They would include your factor 5, 8, and HMWK. Now tissue factor class is insoluble plasma factor. Tissue factor class has no activated form. Then your high molecular weight kallikrein is the activated form is bradykinin. So HMWK, if it's activated, it becomes bradykinin or bradykinin. Then we have your prekallikrein. Activated form class is your kallikrein. In one serine protease, there's a cofactor that binds with it. Tissue factor binds with activated factor 7A or activated factor 7. So again, tissue factor would bind to activated factor 7. Factor 5 would bind with activated factor 10. Factor 8 would bind to activated factor 9. High molecular weight kininogen binds with factor 12, activated factor 12, and kallikrein. The binding of serine protease and cofactors will enhance its function. Now let's talk about the different pathways class. So in your secondary hemostasis, there are three pathways. Let's start first with the extrinsic pathway. Now the intrinsic pathway class is also known as your tissue factor pathway. The reason it's known as your tissue factor pathway is because it is activated class. This is activated when your tissue or there's an injury, which will lead to your factor 3 or your tissue factor to activate factor 7, making it activated factor 7. Now this combination of tissue factor and activated factor 7 will now activate the next substrate. So yung next substrate class is that they would activate factor 9, where factor 8 will bind with it. So again class, it starts with your factor 3. Factor 3 class will bind or will activate factor 7. Now see, itong complex na to, this complex class will now activate your factor 9 with the combination wherein factor 8 will bind to it, which is the cofactor of factor 9. Now factor 9 and factor 8 complex will activate factor 10. And once factor 10 is activated class, this is the start of your common pathway. Together with factor 10, factor 5 will also be activated. So this is your activated factor 10. Activated factor 10 class will now cause it to activate factor 5. Now factor 10 and factor 5 complex will activate your prothrombin, which is your factor 2. And prothrombin will now then be converted to thrombin. So again, let's have a recap class. For the extrinsic pathway, it starts with your tissue factor. Tissue factor will activate factor 7, and the combination or the complex of factor 3 and activated factor 7 will cause factor 9 to be activated. Factor 9 that is activated will now be attached by factor 8, which is its cofactor. Now this factor 9, activated factor 9 and factor 8 will activate factor 10. And factor 10 class will activate factor 5, causing it to become activated factor 5A. And these two class will now activate prothrombin. Prothrombin will now become activated, becoming thrombin. At this time, initiation stage will take place. So again, initiation stage class is wherein fibrinogen will be converted to fibrin by thrombin. The thrombin will be considered as the main enzyme in order for factor 1 to become converted into fibrin. Thrombin will activate factor 13, making it activated factor 13. This will stabilize and will allow for the formation of cross-linked fibrin clot or stabilized fibrin clot. Then the next stage would be propagation. Thrombin will have other factors to be activated. The primary substrate of thrombin will now be fibrinogen. Thrombin will also then activate factors 11, 8, and 5. The activation of more coagulating factors will lead to the generation of more thrombin. Thus, there will be more formation of fibrin that is needed in order to form your so-called stable fibrin clot. Take note, class, that factor 11 doesn't have a cofactor. So that's your extrinsic pathway. Do memorize them. Now, take note of this class, this so-called in vitro or plasma-based coagulation. Now, whenever the blood or the coagulation protein will come into contact with a glass, contact factors will be activated. So, in the secondary hemostasis, we have these so-called contact factors. Now, these contact factors class will be activated by glass. That is why in your laboratory, whenever you would perform prothrombin time and APTT, it is not recommended, class. It is not recommended to use glass sodium citrate tubes because it can lead to the activation of the so-called contact factors. Now, these contact factors class would include factor 12, prekallikrein, high molecular weight kininogen, and factor 11. PK and activated factor 12 will be activated by high molecular weight kininogen, followed by factor 11, which will also be activated. High molecular weight kininogen is the cofactor of prekallikrein and factor 12. Now let's go to the intrinsic pathway class. Now for the intrinsic pathway, this is also known as your contact activation pathway. This is the one that would be activated in the case of contact to glass. Now this would start class with the activation of this one. Let's go back. It starts by being activated by high molecular weight kininogen. So high molecular weight kininogen class will cause the activation of factor 12. Activated factor 12 will now cause the activation of factor 11. Activated factor 11 will now cause the activation of factor 9. And again, factor 9 will be bound with factor 8, causing the activation of factor 10, which is again the start of your common pathway. It's known as a common pathway class because this is where the two pathways would join together. It starts with the activation of factor 10. In summary, extrinsic pathway includes factor 3 and 7. Intrinsic pathway includes factors 12, 11, 9, and 8. In vitro, if in vitro intrinsic pathway, it would include factor 11, 12, prekallikrein, and HMWK. If in vivo intrinsic pathway, only factor 8 and factor 9 are included. Now, the common pathway class would include factor 10, 5, 2, and 1. This is where both the extrinsic and intrinsic pathways would meet. Now, let's talk about the procoagulant's common characteristic. A deficiency generally produces bleeding tendency disorder, except factor 12, precalicrate, and high molecular weight kininogen. So if you have deficiencies class in factor 1, factor 2, factor 4, and so on, you would have a bleeding tendency, except for the following: factor 12, precalicrate, and high molecular weight kininogen. Meaning class, kahit may deficiency ka sa tatlong yan, you will not have any bleeding tendency. Physical and chemical characteristics are known. So the coagulation factors, its physical and chemical characteristics are known. Synthesis of the factor is independent of other proteins. Factor can be assayed in the laboratory. Some of these coagulation factors class are acute phase reactants, such as your fibrinogen, prothrombin, and factor 8. They increase when there is inflammation, as in trauma, pregnancy, infection, and stress. In order to know what pathway is affected, we need to know the importance of these procoagulants. If the patient class has a prolonged APTT, the problem is in the intrinsic pathway. If they have a prolonged PT, then the problem is in the extrinsic pathway. And if you have a prolonged thrombin time, this is a problem with fibrinogen. So do remember this. Now, there are physiologic stages of coagulation. It starts with the generation of your thromboplastin. It starts with the extrinsic pathway, conversion of factor, activation of factor 3 to activation of factor 7 by factor 3. Intrinsic pathway, factor 12 activates 11, 11 activates 9, 9 activates 8. Then the common pathway, factor 10 to 5. This would be followed by the conversion of prothrombin to thrombin, and thrombin will convert fibrinogen into your fibrin. Now, this is a more explanation of the intrinsic pathway class. So the intrinsic pathway again would involve factor 12, precalicrane, high molecular weight kininogen, 11, 9, and 8. Activation of contact factors when they come into contact class with negatively charged surfaces such as glass, kaolin, or ellagic acid. This is not dependent on calcium, so they do not need factor 4 class for this pathway to work. Contact factors are involved in activation of fibrinolysis, complement activation, kinin formation, inflammation, and angiogenesis, measured by APTT. Then we have your extrinsic pathway, which would include your factor 3 and factor 7. Damage to the vessel would result in the exposure of tissue factor on the surface of non-vascular cell. Factor 7 and activated factor 7 would bind to the tissue factor in the presence of calcium. So you need calcium class for the tissue factor to bind, for factor 7 to bind to tissue factor, to form the factor 7, activated factor 7 tissue factor complex, also known as your extrinsic kinase, can activate factor 9 in the intrinsic pathway, measured by prothrombin time, dominant in vivo. Meaning class, this is common inside our body, while your intrinsic pathway is the one that's most usually encountered in vitro because of the exposure to glass, kaolin, or ellagic acid. Then we have your common pathway. This would include factors 10, 5, 2, and 1. Begins with the activation of factor 10 by either the intrinsic or extrinsic pathway. Thrombin clears fibrinopeptides from fibrinogen, forming a fibrin monomer. This fibrin monomer associate in half-staggered overlap pattern between the D and E domain. End result is the formation of a fibrin clot stabilized by your fibrin stabilizing factor or your fibrin or your factor 13. Factor 10 activation by either extrinsic or intrinsic pathway. Common pathway stress is measured by prothrombin time and APTT. So if you have an increased APTT and a normal PT, you have a defect in intrinsic pathway. If you have a normal APTT and increased PT, you have an extrinsic pathway problem. If both APTT and PT are increased, then it's a common pathway disorder. Now let's talk about the roles of thrombin class. It cleaves fibrinogen to form fibrinopeptides A and B and fibrin monomers. It activates factors 5, 8, 9, and 13 as well as protein C. It activates platelets to aggregate and secrete granule contents which generate platelet procoagulant activity. Induces endothelial release of platelet activating factor, prostacyclin, von Willebrand factor, interleukin 1, thrombospondin, tissue plasminogen activator, and plasminogen activator inhibitor 1. Induces granulocyte chemotaxis and adherence to endothelial monolayers and macromolecule. Depresses reticuloendothelial clearance of activated products of coagulation. So these are the different roles of pure thrombin. Now let's talk about the kinin system. This is activated class by both the coagulation and fibrinolytic system. Important in inflammation, vascular permeability, and chemotaxis. Prekallikrein and high molecular weight kininogen are additionally needed to enhance or amplify the contact factor involved in the intrinsic system. Activated factor 12 is in the presence of HK. Converts prekallikrein to kallikrein. Kallikrein accelerates the conversion of factor 12 to activated factor 12. The activation of factor 12 acts as the common link between many aspects of the hemostatic mechanism, including the fibrinolytic system, the kinin system, and the complement system. Let's talk about the complement and the coagulation system. So yung complement class, you're aware of this. These are a set of proteins class that would kill foreign substances in relation to your immunology and serology. Now, plasmin activates C1, causing the cleavage of C3 to C3A and C3B. Now, C3A class would increase vascular permeability. C3B would cause immune adherence of erythrocyte to neutrophils and would enhance phagocytosis. C1 inhibitor inhibits several coagulation factors. The C1 inhibitor class will inhibit complement pathway, and that at the same time, it would also inhibit coagulation factors. Thus, if the coagulation system is activated, complement system is activated as well. If complement is inhibited, coag system is inhibited as well. They occur simultaneously. It can be a complement pathway that is activated because of inflammation, and at the same time, coag system is also activated together with the kinin system. Now, in your coagulation factor, there are three functional groups that you need to remember. The contact factors or the contact group, the prothrombin group, and the fibrinogen group. Now, the contact group would include factor 11, 12, prekallikrein, high molecular weight kininogen. The prothrombin group would include 2, 7, 9, and 10. Fibrinogen would include 1, 5, 8, and 13. Now, take note of the following class. Contact factors are usually activated in vitro, but factor 11 is activated by thrombin. Prothrombin group are essential for the formation of your coagulation complexes, either prothrombinase extrinsic kinase or your intrinsic kinase. Then they are also known as the vitamin K dependent prothrombin group. The reason they are called that class is because they require vitamin K during their synthesis to produce a functional structure. They have structural resemblance to prothrombin, have 10 to 12 glutamic acid units near their amino terminal end. All are serine proteases when activated except protein S and Z, which are cofactors. Activated factor 2 and activated factor 10 are known as your prothrombinase. Activated factor 7 is your extrinsic kinase, and activated factor 9 is your intrinsic kinase. In summary, this is the summary class of the different groupings and the different factors involved. Kindly memorize them. Now let's tackle the contact factor. So this is just a short summary or a short explanation on certain pointers you need to remember for every factor. Let's start with the contact factor, specifically factor 12, also known as your H-band factor. It transforms prekallikrein, a glycoprotein that circulates bound to HMWK, into its active form, prekallikrein. Cleaves high molecular weight kininogen to form bradykinin, activated in vitro. Negatively charged surfaces such as your non-siliconized glass, kaolin, or ellagic acid used in the PTT and APTT acid. This is usually activated in vivo. Foreign materials such as stents, valve, and bacterial cell membranes. So these are the ones activated. So in vitro siya class, outside the body. For it to be activated, it would be requiring the exposure to the following: non-siliconized glass and so on. But for it to be activated inside our body, it has to be exposed to the following: stents, valve, and bacterial cell membrane. Then we have your high molecular weight kininogen. This is a cofactor to factor activated factor 12 and prekallikrein in the intrinsic contact factor complex, a mechanism for activating coagulation in conditions where foreign objects such as mechanical heart valves, bacterial membranes, or high levels of inflammation are present. Then we have your factor 11 found in serum, partially consumed, intrinsic thromboplastin, generating mechanism activated by contact factor complex, thrombin during coagulation generated from tissue factor, activates factor 9. Deficiencies would lead to the so-called Rosenthal syndrome, which would result in mild and variable bleeding. Then we have so we're done with the contact group. Let's go naman to the fibrinogen group. The fibrinogen group, the first one, your factor 5. Now, factor 5 class is extremely labile, deteriorates rapidly, soluble plasma factor, consumed in clotting process. It has a 24 hours half-life. Location is that it is a glycoprotein circulating in plasma, could also be present in the alpha granules of platelets. Platelets would release partially activated factor 5 at the site of injury as a cofactor to activated factor 10. Accelerates thrombin generation more than 300,000 times compared with activated factor 10 alone. Initial small amount of thrombin generates the first conversion of factor 5 into its activated form. Regulation is done by thrombomodulin-bound thrombin, which would activate protein C. Protein C class inactivates factor 5. Both activated and then ultimately inactivated by the generation of thrombin. Then we have your factor 8 or the antihemophiliac factor. This is an acute phase reactant consumed in clotting, not found in serum. Produced primarily by hepatocytes, also by microvascular endothelial cells in the lung and other tissues. Free form is unstable in plasma. Circulates bound to the von Willebrand factor. Now, this is activated class when thrombin clears factor 8 from von Willebrand factor and activates it. Activated factor 8 binds to activated platelets and forms the intrinsic kinase complex with factor 9, activated factor 9, and calcium. Also inactivated by protein C, same with your factor 5. Production is covered by genes carried on the X chromosome. Deficiency is involved in severe bleeding and symptoms associated with hemophilia A. Deteriorates more rapidly than other coagulation factors in stored blood. Todd component plasma levels drop to approximately 50% after 5 days. Now, your factor 8 class has different nomenclatures. Usually, you would be wondering why they have different nomenclatures, because remember, class factor 8 has a lot of function, so thus it has different nomenclatures. Kindly memorize them. They may come out in your quiz. And the last group, the fibrinogen group, we have your von Willebrand factor. Now, von Willebrand factor class is an acute phase protein, similar with factor 8. This is a large multimeric glycoprotein. Has multiple subunits of 240,000 Dalton, each produced by endothelial cells and megakaryocyte. Combines to form multimers that range from 500,000 to 20 million Daltons. Function is responsible for platelet adhesion and the transport of factor 8, stored in alpha granules and platelets and in Weibull pallid bodies and endothelial cell. Endothelial cells release ultra-large multimers of von Willebrand factor into plasma, degraded into smaller multimers by a VWF cleaving protease. ADAMS13. ADAMS13 stand for a disintegrin and metalloprotease with a thrombospondin type 1 motif member 13 in blood vessels with high shear stress. Severe bleeding and symptoms associated with abnormalities in von Willebrand molecular structure and concentration. An inherited disorder with several described mutations. Levels of von Willebrand factor vary by ABO blood type. So nagbabari siya depending on your blood type class. If you are a group O individual, you tend to have lower levels of von Willebrand factor. It is also an acute phase protein as well as factor 8. Then we have your factor 1 or your 5 nanogen. This is a large stable with a molecular weight of 340,000 Daltons synthesized in the liver. Usually has a concentration in the plasma of 200 to 400 milligram per DL. Most concentrate. Function is that it is the primary substrate of thrombin. Converts fibrinogen to fibrin monomer, then into a polymerized fibrin clump. Essential for platelet aggregation. It links activated platelets through their glycoprotein 2B3A platelet fibrinogen receptor, an acute phase reactant protein. Level increases in inflammation, infection, and other stress condition. Platelet alpha granules would absorb, transport, and release abundant fibrinogen. So that ends the lesson 3. Here are your references. Thank you.